We begin investigation 3 by looking at how normal and cancer cells divide. We're beginning by looking at normal cells, here with these yellow gram cubes. Every 15 seconds, we have them divide by adding 2 more cells. And what we'll see is that as we add the cubes or the cells to the beaker, that at a certain point, they cover the bottom of the beaker and they stop dividing in this model. This is known as contact inhibition, and many normal cells do this. Unfortunately, most cancer cells do not show contact inhibition, but rather divide and can form tumors. So the white cell, in this case, indicates one cancer cell in a mix of normal cells. After 15 seconds, we added another cell, showing that it divided. And then those cancer cells divide, and we add 2 more. 15 seconds later, we're now adding 4. 15 seconds later, 8, indicating that each one of the cells divides. And finally, at the end of another 15 seconds, we'll add 16. Now clearly, these cancer cells are not exhibiting contact inhibition. They're growing, they're forming a mass, and this mass is called a tumor in the case of cancer cells. Now up to this point, we've discussed cancer, but the students haven't actually seen any cancer cells. From this point on, they're now going to have this opportunity. We're going to look at lung and breast tissue. First, lung, and we're going to just review what normal human lung looks like. And we saw this in investigation 1, so this is just a review. We're only going to go up to 40 objective, in this case up to 400 total magnification. And here's the normal lung tissue. And we discussed before the importance of the alveoli and how it is at this site that gas exchange occurs in the lung. So this normal lung structure has to be in place in order for the organ to work. Once again, we record our observations in the student data record. Again, we always lower the stage before removing any slide, which is what Philip's doing here. Okay, now we'll take a look at a section of human lung cancer. And what we're going to see is the loss of the normal tissue structure and the replacement by a massive amount of cancer cells. So you don't see the alveoli. In fact, the alveoli are crowded with these undifferentiated cells. So it's very easy to see why normal function of the lung would be compromised by lung cancer. And we'll record this in the SDR as well. Next, we're going to look at human breast tissue, and we're going to look at normal human breast tissue first, and we did this in investigation 1 as well. Actually, we'll be looking at inactive normal breast tissue in this case. In all of these observations, we're only going to go up to a total of 400 magnification. That is only up to the 40x objective. Okay, so here's that view again of normal, inactive human breast tissue. You see the connective tissue here in pink. We also see the adipose cells, or the fat cells, in the tissue. And then the lobe structures as well, stained in a darker purple in this case. Okay, so that serves as a review of normal human breast tissue, and we'll once again record this. Now we're going to take a look at human breast cancer. And in this section, you can see these massive lumps of undifferentiated cells that have divided, with essentially no control from the cell cycle. Now, fortunately, these lumps can actually be felt upon self-examination or examination by a physician. While wonderful advances are being made now in the treatment of breast cancer, the success of such treatments almost always depends on detecting these lumps at a very early stage. This will end our third investigation of cell cycle and cancer.